talk about the Benedict test. So Benedict test is a test that is a qualitative test. That means it detects if there is any sugar, glucose is present or not. So before coming in detail with the Benedict test, we must know what is actually the reducing sugar, because Benedict test gives the positive result for the reducing sugar. Okay, so what are reducing sugar? Carbohydrate is divided into two forms: reducing sugar and non-reducing sugar. Non-reducing sugar. Okay. So what are reducing sugar? Reduction, oxidation, reduction. There are two processes. Reduction, that means addition of hydrogen ion, addition of electrons, and emission, donor of the, if someone gives oxygen, that is also reduction. And oxidation is the opposite thing. Addition of oxygen, emission of hydrogen, emission of electron. So this is actually the oxidation and reduction. So whenever any substance is oxidized, that means oxygen is added. That means the another one, the opposite side, there they get reduced. Suppose this a sugar that are called reducing sugar. That means they reduce the another component. They reduce another component. That means they are oxidized. They oxidize itself and they reduce the another component. So that's why they are called reducing sugar, as because they are reducing the another components. So they are reducing agent. Reducing sugars are the reducing agent that reduces the another one, and they get oxidized. They itself get oxidized. So that is the reducing sugar. In carbohydrate, all the monosaccharides are reducing sugar. All monosaccharides. All monosaccharides are reducing sugar. Okay? And all polysaccharides. All polysaccharides are non-reducing sugar. Okay. Disaccharides, for the disaccharide, except sucrose. Disaccharide, I am writing here. Disaccharides except sucrose. Maltose, lactose, these are all reducing sugar. So reducing sugar must have free aldehyde group. So all these monosaccharides have free aldehyde group. Disaccharides except sucrose, because sucrose doesn't have any free aldehyde group. So if any carbohydrate, especially monosaccharides, have free aldehyde group, that are reducing sugar. They can give the hydrogen to the another component. Okay, and they get oxidized. That means they are giving hydrogen, so they get oxidized, and another one they are reducing. Okay, so that's why they are reducing sugar. Now come to the Benedict test. So Benedict test is a test that use the Benedict's reagent. What do we call Benedict's? Benedict's. We must put S here, because Stanley Rosseter Benedict, the name of the scientist who invent the Benedict's reagent. A reagent that is actually invented by these scientists, Stanley Rosseter Benedict. That is the Benedict's reagent. What are the components of Benedict's reagent? Copper sulfate, sodium carbonate and sodium citrate. So these are the main components of the Benedict reagent. Sodium carbonate makes the solution alkaline. Sodium citrate prevents the precipitation of the cuprous hydroxide, and copper sulfate, here it is present as a cupric ion, and what happens whenever we add the reducing sugar, it converts the cupric ion to cuprous ion. That sugar reduces the copper sulfate, that means it converts the cupric ion to cuprous ion. So if it's a reducing sugar, is a glucose, alkaline medium, it converts it forms the enediol by tautomerization. Now what is tautomerization? I must explain here. This is a keto group. Okay. And this is aldehyde. This is a keto group. This is a aldehyde. Keto or enol group. Ester. If there is ester, then I would call it as the enol. So keto, when transferred, it shift to enol. That is called the tautomerization. So whenever we are adding reducing sugar to the Benedict reagent, these are the components. Alkaline medium. That alkaline medium is actually made by the sodium carbonate. So glucose by the alkaline medium, it transforms into enediol. Enediol is a strong reducing agent that reduce the copper sulfate, that means the cupric ion to cuprous ion. Now look, the product that is formed initially by the reduction, that is the copper hydroxide. This is copper hydroxide. Now what happened? One molecule of water will be discharged by this copper hydroxide. This copper hydroxide is yellow in color. That not precipitated. That yellowish substance is not precipitated. Why? Because of the sodium citrate. That sodium citrate prevent the precipitation of the copper hydroxide. Now what will be then the next incident, what will be occur? Now look at here. Copper hydroxide, CuOH, if I do, okay, so CuOH, now remove H2O, remove that part. What will be the product? Cu2O. Okay, so that is the cuprous ion. Okay, that is cuprous, copper hydroxide to cuprous oxide. Okay, the color of the cuprous oxide is brick red. What should I write? Yeah. Copper oxide is a brick red in color. Whenever the solution turns brick red in color, that indicating that there is a reducing sugar. Okay. So this is a qualitative test. Qualitative test means it's denoting that if any substance is present or not. Okay. So that is the qualitative test, and that's all about the Benedict test. If my video make you understand, definitely subscribe my channel, and for the next time we will meet on next topic, new video. Till then, thank you.